Hello, welcome back to my practical class. In this video, I will show you how to calculate the GC content of a DNA sequence using Python. This is one of the most basic and important concepts in bioinformatics. I know someone may ask, what is GC content? GC content means the percentage of nitrogenous bases in DNA that are either guanine or cytosine. The DNA has 4 nitrogenous bases, which are adenine with letter A, thymine with letter T, guanine with letter G, cytosine with letter C. And adenine always pairs with thymine. Guanine always pairs with cytosine. So the word I use for it is at GC. At GC, A to T, G to C. So when we talk about the GC content, we are only concerned about the percentage of pairing between the guanine and the cytosine. And I know someone will ask again, why does it matter? Why are we looking for the GC content? It's because the GC content determines DNA stability. How stable the DNA sequence is as a result of the GC content. The GC content also determines gene region. So any area that has enough GC content, it means there is a particular gene in that area. The GC content is also used to study evolutionary differences among organisms. Closely related organisms tend to have similar GC content, while organisms that are not related tend to have dissimilar GC content. So GC content determines structural stability because the hydrogen bond between the GC is 3 pairs, while the hydrogen bond between the AT is 2 hydrogen bond. The GC content also determines the melting temperature. Higher GC content increase the temperature required to denature the DNA double helix. The GC content can also be used in the lab when it comes to PCR design and primer design. In polymerase chain reaction, the GC content of a primer must be carefully balanced. So when you want to design a primer in PCR, you must consider the GC content of that DNA sequence. Now, this is a standard formula for GC content calculation. So GC percentage is equals to count G plus count C all over total length of DNA times 100. So this is the standard formula to calculate the GC content. So this is the formula we are going to use in our Python to calculate the GC content. These are some of the examples of organisms that their GC content have been calculated from their genome. We have the Homo sapien with 41% GC content, yeast 38%, Tacres with 36%, E. coli with 50% GC content. Now let's move to a practical session where I'm going to show you how to calculate this GC content step by step using Python. Okay, now let's move to a practical session where we calculate the GC content of a DNA sequence using Python step by step. I believe by now you have Python in your system, but if you don't have Python in your system and you want me to do a video on how you can install Python and set up the Python environment successfully in your computer system, please comment on the comment section Python needed so that I can do a video on how you can install Python successfully in your computer system. So this is my desktop. So I will go to the search button, then type Python because I have already installed Python on my system. So type Python, you'll see it will show with the version Python 3.14. That is a Python that was installed in my system.
then click on it, then the Python environment will open. So this is where you write your programs, this is where you write your codes, this is where you compute everything. So the first thing we need to do is to declare a variable. This is a Python environment. Python is a very user-friendly programming language, easy to understand, simple to use. So let's declare a variable. Okay, so we have declared our variable. We say DNA is equals to AT, that is our sequence, our DNA sequence. It could be anything. You can add any sequence of your choice. It could be the sequence of insulin, any sequence at all. You declare it. Then we will come to under. We now say G is equals to DNA.count G. So we are telling the Python to count, to check the DNA sequence and count all the letter G. How many are the letter G? Then we also declare another variable which we said C is equals to DNA dot count C. That is, we are telling the Python to also count all the letter C. Then we now say the GC content is equals to G plus C divided by the length of the DNA times 100. That is our formula. That is what we are telling the Python to do. We said, okay, to get the GC content, this is what you will do. Add the total number of G to the total number of C in our DNA sequence, divide it by the length of the DNA, then times it by 100. Then we use the print statement, print GC content. We have already described what GC content is. So we have told the Python to print it. So the Python has successfully given us the GC content for our DNA sequence, which is what? 60%.
In the next video, I will show you how to generate the reverse complement of a DNA sequence using Python. If you are learning bioinformatics, this is the foundational knowledge. If you find this video helpful, please subscribe and ask and drop your question in the comment section.